Today I will present slides about IP3 DAG pathway. It is GPCR mediated or G protein coupled receptor mediated pathway, and the full form of IP3 is inositol triphosphate and the full form of DAG is diacylglycerol. So we all know that GPCR is a 7 transmembrane domain or serpentine receptor, and here alpha, beta, gamma, these heterotrimeric proteins or subunits are bounded with each other. So what happens in inactive state? No ligand is bounded with this GPCR and GDP is bounded with the alpha subunit and it is in inactive state. But when ligand comes and binds with this receptor, then what happens? Then GDP converts into GTP by the action of GEF, and then this GTP binds with the alpha subunit. And what happens? This beta gamma subunit and alpha subunit breaks and separates from each other. Alpha is in monomer form and bounded with GTP and is in active state, and beta gamma is in dimer form and separated from the alpha subunit. Then what happens? Then this activated in IP3 DAG pathway, what happens? The activated alpha subunit then binds with the PLC or phospholipase C, especially the phospholipase C beta. Then after that, this activated PLC then binds with the PIP2 or phosphatidyl inositol diphosphate, which is then activated and separated in two parts. One is hydrophobic and lipophilic DAG diacylglycerol, which is bounded in the cell membrane, and another one is hydrophilic IP3 or inositol triphosphate, which is water loving. So it releases, and then what happens? This DAG is bounded with protein kinase C, but protein kinase C is not activated. But IP3 then enters into the cell membrane and binds with the cell membrane of the endoplasmic reticulum. And after binding IP3, endoplasmic reticulum releases calcium ions readily. After that, what happens? The surrounding calcium concentration increases, and these calcium ions then comes and binds with this PKC DAG unit. After binding with the protein kinase C, what happens? This protein kinase C becomes activated, and after activation, this protein kinase C then activates the target protein, and after that phosphorylation happens and then cell response happens. So coming to the next part of the summary, we will get that in IP3 DAG pathway, what happens? At first, GPCR is not activated when GDP is bounded and there is no ligand present. Then what happens? Ligand comes and binds with this serpentine receptor or GPCR. Then GDP converts into GTP and binds with the alpha subunit and activates it, and alpha is separated from the beta gamma units. And what happens? Then activated alpha in IP3 DAG pathway, what happens? Alpha then binds with the PLC or specifically the PLC-beta, phospholipase C beta, and then activates it. And then this activated PLC-beta activates PIP2 or phosphatidyl inositol diphosphate. Then this phosphatidyl inositol diphosphate breaks into two parts. One is hydrophobic and lipophilic DAG or diacylglycerol, and another one is IP3, which is water loving or hydrophilic. And this IP3 then what does? IP3 then binds with the cell surface receptors of the endoplasmic reticulums and then activates it to produce calcium ions readily. And then endoplasmic reticulum releases calcium ions readily, and the surrounding calcium concentration becomes higher or greater, and these calcium ions then enters into several processes including camodulin, sperm motility, egg activation, and it also enters into this IP3 DAG pathway. And what does? Protein kinase C is binded already with the diacylglycerol or DAG, but not activated. When calcium ion comes and binds with this unit or DAG-PKC unit or protein kinase C, then what happens? PKC becomes activated, and then this activated PKC activates the target protein, and then phosphorylation happens, and then cell response or further cell response happens. So in cell surface receptors, through GPCR or G protein coupled receptor, we can get two pathways. One is cyclic adenosine monophosphate pathway or cyclic AMP pathway, CAMP pathway. Another one is IP3 DAG pathway. So in CAMP pathway, what happens? GPCR binds with the ligand and then G protein activates. After that, it activates the AC, and this AC activates the AMP to produce CAMP readily, or ATP to CAMP. After that, this CAMP binds with the regulatory parts of the protein kinase A, and then regulatory part and catalytic part of this protein kinase A breaks from each other. Then this catalytic part enters into the nucleus from the cytosol, and what does? And then this activates the CRAVE, which is already in nucleus, and then this activated CRAVE becomes activated and does further responses including transcription, translation, and cell response. In the other hand, in IP3 DAG pathway, what happens? GPCR binds with the ligand, becomes activated. After that, alpha or G alpha is activated. Then it comes and binds with the PLC-beta or phospholipase C-beta, activates it. After that, PLC-beta activates PIP2 or phosphatidyl inositol diphosphate. This phosphatidyl inositol diphosphate or PIP2 breaks into two parts. One is DAG and another one is IP3. DAG is already membrane bound and IP3 is a hydrophilic. So IP3 comes and binds with the receptors of the endoplasmic reticulum, and then endoplasmic reticulum releases calcium ions. These calcium ions comes and binds with the protein kinase C, which is already bounded with the DAG. After that is activates PKC, and this activated PKC then what does? Activates protein target proteins and phosphorylation, and further cell responses or activations happens. So this is the short summary or description about IP3 DAG pathway. Hope you like it.